eta ongi etorriak izan zaitezte, konferentzia hitzaldi ziklo honetara. Eta konferentzia ziklo honi deitzen diogu, eta urte honetatik aurrera izango da, udazkeneko jardunaldi kulturala, 2023, hitz egin dezagun zientziaz eta teknologiaz. Eta hemen 5 hitzaldi izango ditugu. Lehendabizikoa, gaur Nico Leischek emango diguna, eta beste 4 hitzaldi egongo dira horren segidan. Abenduaren azkeneko izango da, abenduaren 20ean. Eta gainera nik uste abenduaren 20ko oso interesgarria da, zen orain grabatu dute serie bat, telebista serie bat, Nikola esaten dut, serie bat, 13 kapitulukoa, eta pelikula hori edo serie hori da Territorio Gravedaz izenekoa. Eta aktriza edo aktoreak emango digu kosmologiari buruz berak ikasiko duena. Hori izango da, ziklonen amaiera hitzaldia. Berriz ere esaten dut, ongi etorriak eta parte hartu ezazue hitzaldi guztietan. Buenos días a todas y a todos por venir o acercaros a este ciclo de conferencias que de este año estamos llamando Jornadas Culturales de Otoño 2023, hablemos de ciencia y tecnología, en el que vamos a tener 5 charlas de ámbitos diferentes, de áreas diferentes. La primera es la que nos va a dar el doctor Nico Leisch, de la Universidad de Heidelberg, y a partir de la misma vamos a tener hasta el finales de diciembre otras 4 charlas. La última, y quiero comentar que será el 20 de diciembre, es una charla interesante porque será la única en la que no una persona investigadora que conozca el mundo va a tratar, va a hablar en esa conferencia. En este caso, la persona que va a hablar va a ser una actriz. Y esta actriz es la protagonista de una serie cinematográfica que pronto se pondrá en las pantallas, que se llama Territorio Gravedad. Entonces, ella nos hablará, una actriz, acerca de cosmología. Bienvenidos a las charlas y espero veros a todos vosotros y a todas vosotras en este ciclo de conferencias. Y ahora le voy a dar el paso a Ivonne Cancio, profesor de esta universidad, que va a hacer la presentación del conferenciante Nico Leisch.
Vale, ni oso laburra izango naiz, eskerrak etorri zaretelako, seré muy corto, muchas gracias por venir. Y bueno, voy a utilizar la lengua de Darwin para hacer la presentación de Nico. So, I am a professor in the University of the Basque Country, and for me it's one of the biggest honors that I could have this year, 2023, to have been the responsible to host one of the biggest expeditions of this century, which is the TREC expedition. It's an idea that was launched by the European Molecular Biology Lab, which is based in Heidelberg, very far away from the sea. 50 years doing molecular biology and advanced microscopy, and they have decided that life happens outside the laboratory. So they are coming to the field where life happens in context. And Nico is in charge of bringing all this big equipment, all these big Ferraris, into the marine station of the University of the Basque Country in Plentzia. So we have invited him, Nico, who is the responsible of this advanced mobile lab that we have in Gorlitz Park right now, to talk to you about what's an advanced mobile lab and what you can be doing in an advanced mobile lab. Mind you, they are not only coming to do science, they are also coming to do outreach. And we have outreach events associated to what Nico and his colleagues are doing in the lab. So stay tuned, because there are things happening in Bilbao, in Plentzia, in Gorlitz, and if you are willing to have information, just come on and ask me afterwards. But now is the moment for the talk of Nico. He's a very easy man to reach to, a very nice man to have besides. That's very important in science. Touching people, it's very important, because love happens and science happens when you touch people. And Nico is very touchable. So Nico, thanks for coming, and you know, the students of the University of the Basque Country are listening to you. Okay, thanks.
Thank you very much, and hello everyone. I'm going to switch a little bit the presentation. Typically the acknowledgement come all at the end, but I want to start this presentation with a big thank you to the university and especially to Ibon and all his colleagues for hosting us, for welcoming us, for showing us the beauty of the Basque Country. And here you can see the beautiful view we have every day from our mobile lab in Gorlitz, right next to the marine station, where we are working. And I mean, you cannot but smile every day when you're starting your day with such a view. Now, who am I, why am I talking to you today, and what are we doing? So I'm trying to cover all these questions over the next half an hour, and I'm also happy to take any questions and discuss with you about all of this. Now, I'm part of the EMBL, or the European Molecular Biology Laboratory, which is an intergovernmental lab that's doing life science. It's funded by all of Europe and then a few more member states. We have stations throughout Europe, and we basically belong to all of you. And following this concept, we are now also touring through all of our member states and also visiting you and want to do science with you. Skip this one. So we're not only doing research, but key missions of our institute is also to provide scientific services, so providing training, generate new methods, and also what we call here integrating life sciences, so building communities. And so it's not only to come here and do great science, but it's really also to get in touch with you, to discuss with you, and also to offer what we bring to the research community on site. We're working in 5-year cycles of big plans, and with the newest plan, it's called Molecules to Ecosystems. As Yvonne already mentioned, life does not only happen in a test tube. We can learn a lot from all the work that we do in a test tube, but it's ultimately out there in nature where we need to understand, is what we find in the test tube representative for everything, or is it a special case? And of course, especially with test tubes, if you have a single organism in isolation, it's very difficult to understand what its behavior is, what its life cycle is, and so forth, because you're missing all the context. You're missing all the things that you interact during its normal lifespan. And so this is really central to our new project, is that we're really studying nature in context with the environment. At the same time, we're also realizing we as a society, beyond the scientific community, also are challenged. We are facing things like climate change, pollution, loss of biodiversity. So these are really urgent topics where we also need to address and tackle them. And most importantly is really, again, collaboration, collaboration, collaboration. Especially the expedition that I'm going to show you afterwards can only happen because so many people work with us, be kind to open the doors and host us and collaborate with us. Now we started this project, and what we called it is planetary biology, so to really look at it on a global scale, zoom out of the lab, take the environment into account and look at it on a global scale. And we want to, because also from all the technical revolutions that we saw in the last 10 years, if you think about sequencing and so forth, we can really start combining traditional lab methods like heavily based molecular biology with more ecological approaches, evolutionary approaches, and all the geoscience approaches. And so we're going from the field to a controlled environment, and as a next step is really then also to start modeling these interactions. If we're looking at the challenges, of course, the big one nowadays is climate change. We're having the warmest summer on record, and it's going to be the warmest summer on record this century so far, because we are set to break these records again and again. We're looking at chemical pollutions. We're more and more aware of how things have long-lasting impacts. For example, right now in the news is a lot of discussion of forever chemicals. We're facing antibiotic resistance spread from agriculture, hospitals, but we also see how these antimicrobial resistant genes can flow through the environments. And of course, the big one, biodiversity loss, especially when we're thinking insects, bees, and so forth. Now, what are we actually going to do about this? So the first thing we need as scientists is data. And so the big step for us now is to go away from our lab where it's comfortable and go out in the field where it's uncomfortable. And we decided to make a big expedition throughout the European coastline. That's why we called it a TREC, Traversing European Coastlines. And we're focusing on the coastal regions because they're extremely important habitats. More than half of the European population lives in coastal territories, so it's not just for holidays, but it's really where we live. We see a lot of biodiversity. It's where the land and the sea meets. Wherever two habitats meet, there's a lot of interface, which is interesting because there's different niches, there's different forms of adaptations happening. And it's also a habitat that we are strongly impacting with big cities, with the outflows of our rivers, agriculture, and so forth, is directly impacting that. Last but not least, often these habitats also studied under one aspect or the other, so either from the land side, from the sea side, from the freshwater side, and so forth. So our aim was really to come up with a holistic sampling scheme that integrates all of them so we can get a broader picture of it. And EMBL is not a national funded research institute, but a European funded research institute, is also at a perfect position to lead such a European-wide sampling expedition. And what we are doing is really to standardize and do the same thing throughout all of Europe in order to really have a European-wide data set that we can then compare broadly. The coasts also allow us to pick up on interesting gradients. If we're thinking natural and human gradients, we have high amounts of population, especially in the Mediterranean area, versus less populated area the further north you go. We have strong salinity gradients, temperature gradients, tidal influences, and so forth. We are looking at this from throughout all the organismal scales. So we have projects that are working on the smallest viruses. We have projects that are working then on multicellular organisms, plants, animals, and so forth. And really we're looking at different aspects in this project, from function of single molecules, for example, photosynthesis pigments, all the way to how single species behave, how their development plan is, all the way to complex communities throughout Europe. And we're looking at it a little bit across timescales, looking at, for example, behavioral reaction, which can be in milliseconds, all the way to geological timescales. Now, what are we actually doing? Right? So we're sampling along the European coastline, and what you see here is a beautiful map, and all the yellow dots are actually sampling sites, about 120 throughout all of Europe. And we've split them based on what type of sampling site they are. Ideally, we have some pristine nature, or as pristine as it gets in Europe. We have agriculturally impacted sites. We have cities and ports, and as well as river outputs. So we're spanning a broad spectrum here as well. And then we're deploying a fleet of mobile labs to actually do all of these samplings. But of course, land is only one part, so we've partnered up with Tara Oceans. If some of you might be familiar with it, it's been the Tara Oceans expedition has been going on for more than 15 years now. It's been very successful, and it's been very standardized sampling throughout the world's oceans, and a lot of our modern understanding of oceanography is really based thanks to their pioneering work. As of this now, it's also been sighted off Borneo, correct? And they will be here for the next 2 weeks, first working with us for a week, and then also doing a week of public outreach around here. So if you're interested, also keep your eyes out for this. And of course, the most important pillar for us is really also the network of marine research institutes where we can connect with, not only for the infrastructure, but much more importantly also to discuss with the local scientists, because you know your coast best. And you have experience, you know where to work, you know how to work, and so by, again, forming a community, this will inform the whole expedition to much more. Now, this is how a typical sampling day looks like for us. So we're going on the to the shore, and we're working from inland, so areas that are not affected by marine waters, all the way into shallow waters. This is the part that the land team can take care of. And then offshore, we have TARA that are doing the similar sampling offshore. And we're taking everything from topsoils, superficial sediments, shallow waters, and so forth. And each point on this map is actually representative for a transect. So we're taking a 40 meters long transect. We're taking then subsamples throughout this transect. And just if you want to have a small glimpse into then what the protocols are, it starts from very generic description of the site, taking pictures, taking GPS coordinates, saying, do we have vegetation here or not? Is it trees? Is it dunes? Is there no vegetation? Then starting with collecting plants around it, collecting soil samples, and then the real work starts to split up the soil samples, preserve them for all the different downstream analysis, being heavy metals, pollutants, DNA extractions, RNA extraction to cover the whole omics suite, organic carbon, and so forth, as well as microscopy and samples for future analysis so that we don't have to necessarily redo this whole expedition next year again. We're also banking some samples for any or all protocols that will come up in the next years, new technologies that can either more clearer or address open questions or come up with completely new technologies. We're doing the same thing in the intertidal, where we're then also going out with so-called Niskins bottles, where we're sampling water, and then again doing the same in-depth characterization of this site. And so with this, we're getting really a good overview of the metadata and the characteristics of this site. Everything from inorganic components all the way to meta-barcoding to know, for example, about the microbial community, but also then metagenomics and transcriptomics to better get an understanding on the genomic level who is there, and on the transcriptomic level, what are they doing, how are they interacting with, and so forth. Next step, we're also cultivating, where possible, selected species that are important for these ecosystems, be it sponges, macroalgae, and so forth. We're also characterizing the habitats, for example, using passive acoustics to record the underwater soundscape and derive fish populations from there. And for me, amazing and fascinating and a very work-intensive project, for example, is paleocores, so taking long sediment cores where possible in the marine environment, and then slicing through them centimeter for centimeter. And as these sediments are very stable, we can actually go back in time. And so we have 100 to 150 years within a meter of sediments, so we can actually go back and see, for example, where pollutants accumulated. And we can also get information back from previous, especially bacterial communities from the past, where we can, for example, also then try to revive persistospores from past communities and really have a window into the biological past on sites. All of this gives us a lot of data. Just outlining a few here on the bottom, from omics data, microscopy, to metabolomics, physical parameters, chemical profiling, and so forth. And this is also where we say, okay, we are quite a few people, but we will never be enough to work with this data set. And so EMBL is committed to openly share all of this data that we are collecting throughout the expedition, make them available to the whole European and the whole worldwide research community, because continental data set needs also a big effort to analyze, and open data is the only way to move forward on this. Another fascinating topic is also chemical profiling, especially when it comes to let's call them nasty compounds, anything from pesticides, fluorinated compounds, heavy metals and so forth that can negatively impact us. And one of the really interesting projects here is really looking at pesticides, which microbial organism can break down pesticides, and also what's the effect of breakdown products. Typically, pesticides are measured for the exact molecule, and as soon as you split off, for example, a hydroxy group from the molecule, it's no longer detected at the certain peak, and so the molecule for this assay is gone from the environment. Now, I think we can all agree that that's not really how biology works, and so we don't have a good understanding what these degradation products are actually doing on biological life, and we also don't know how much they accumulate. And so one of the big projects within this expedition is really to look specifically for who can degrade which pesticides, and what do those degradation products then have an effect on other biological entities. This is how the everyday life looks like for our samplers. This is the land-sea interface team, so those that drive throughout all of Europe and keep sampling, come day and night, come rain or hail. No, we're not making them work during hail, no worries. But just so you get a little bit of an idea of what it means, it's of course always nice when you get some visitors on the sampling sites, but sometimes it can also be quite tough when you need to wade into the shallow waters in order to collect the water samples. And you get a lot of questions when you start doing these kind of works on the beach, especially when it's tourism season. Now, the whole TREC expedition was initiated by EMBL, but we also very soon opened the doors for any researchers throughout Europe that was interested to actually piggyback on our expedition, especially if it meant just a little sampling effort to cover also a project. So for example, we call them these plug-in projects. So for example, they can be collect a little sample so we can assess microplastics throughout Europe. That's easily done, and it benefits the researchers, and it benefits us because it covers a new part within this project. However, not all of these projects can be as easily done as just collect another little tube and freeze it. And so for this, we really need mobile labs. So, and this is also my core responsibility. And what we want to do with this, obviously the mobile lab needs to be mobile. That's the easy one. And what we want to do here is to bring advanced technologies into the field. So we've tried to sit down and think of which technologies are not always available on site, which ones make a difference. For example, it's easy to take a soil sample, put it in an Eppendorf tube, freeze it, and then sequence it a couple of months later. You don't need much sophisticated equipment for that, in best case, a freezer. So we were thinking, okay, what can make a change or a difference? What enables us to have new insights into biology? And importantly also, let's always move the same machines throughout Europe so we can standardize. So we're always looking at the same way with the same machines, with the same protocols, so we can compare these things afterwards. And last but not least, these mobile labs are also great support platforms for example, public outreach, but also scientific workshops to train people on methods and so forth. And machines and cars are one part of this, but it also takes people. And so I would like to briefly introduce you here to my team of experts in various technologies that are actually with me traveling throughout Europe. They said goodbye to their families for the next 6 months and are just traveling like a circus through Europe and enabling the TREC science and the local science on site. So this is the crown jewel in our fleet. This is the so-called advanced mobile lab. This is how it looks like when it's driving. It's basically a trailer on the heavy side, but that's it. But when you're inside, it gets a bit tight. So when we're on site, we can actually expand the sides and nearly triple the working space within our mobile lab. And this is how it looks like right now, parked in front of the institute in Görlitz. And overall, it easily accommodates about 10 people without anyone stepping on anyone's toes. We have all the basics needed for molecular lab work in there. So all the infrastructure that you need. We also have, for example, a dedicated sample area where whatever we bring in wet, dirty, and so forth samples, we can work with them first before we go to the more sensitive machinery. And so the advanced technologies that we said, okay, this can really make a difference, is on the one hand preparational techniques for electron microscopy, especially cryo techniques, so where we can freeze biological samples in like this. So within 20 milliseconds, we can arrest every biological process within these cells or organisms. And we're seeing now completely new level of insights in cellular preservation from unicellular organisms all the way to complex organisms, where we see new organelles, where we see where we see associations that we haven't seen before. We can preserve bacterial symbionts and so forth. And so it's really giving us a new appreciation for the biology on site. We're also using automated microscopy to, first of all, optimize the little time that we have on site and to really look at complex environmental samples as they come in and then be able to first get good overviews, but then also identify the organisms that we are interested in and then start more automated high-resolution imaging, be it time-lapse imaging or three-dimensional imaging. And last but not least, the key to a lot of these workflows is our imaging large particle sorter. So this is a particle sorter that can sort organisms up to 500 micrometer in diameter, which is already huge in biological scales. And additionally, it takes an image of every single particle that it sorts. So we don't only have the classical flow cytometry parameters, but we can also then use the images generated during the sorting process to further redefine the gates that we generate here and really go from complex, diverse samples to ideally pure cultures of pure enrichments. With this, we can cover a lot of workflows, imaging, cultivation, sorting, any downstream omics, and so forth. But what was important is also that all these machines can work with each other. So, for example, we can sort first, then image with the microscope, and then preserve the exact same sample for electron microscopy. So we really get a correlative data set and as many aspects as we can cover about certain organisms. We can also use the confocal microscope to actually label the cells that we're interested in and then sort them out with the sorter and then go into downstream applications and so forth. And the big challenge that we have when working with environmental sample is just the huge diversity. If you have a look at it, it's like a wildly diverse soup where your organism that you're interested in is in there, but you need to find a way to getting it out. And what previously you hired unpaid interns and they were sitting there for weeks and weeks and weeks and picking the cells that you want, and now we can automate a lot of this. I would like to take also the chance here to briefly show you one of the projects that's going on, which is going on actually right now as we speak. This is the so-called Team Plan XM. It's a diverse group and also a very collaborative group, like many of the TREC projects. And they are looking at ultrastructure of plankton. And they're using a technology called expansion microscopy, which is a method that came up in the last few years. They're focusing both on environmental samples, but also connect to local culture collections on site. And what they're doing is, because they want to image small unicellular organisms, and that's difficult with microscopes because you need very good resolution. But there is a trick that they apply. Instead of buying a more expensive microscope to have a better resolution, they actually just make the organism bigger. So you can, by introducing the organism into a hydrogel, then dissolving everything that keeps it from scratching, you can then swell this hydrogel and expand your organisms about 4 times in size in all 3 dimensions. So it's just like the reverse shrink ray. Once that it's done, you have multiple benefits. It's easier to image. You need less of a high-resolution microscope, but also many more targets within your organism actually accessible if you want to label them with either dyes or specific antibodies. And so we're getting a much higher success rate after the expansion to actually label the structures of interest. And so what you can see here as an example, this is then a 3-dimensional data set that you get afterwards. We have here in turquoise is the DNA, in green is the cytoskeleton, and then in red is just generic protein content. And also another one here, which is even more spectacular. Looking yellow is lipids, and then we're going to be in pink, we're going to look at tubulin. And so we can really get a 3-dimensional appreciation of these organisms. And also there we're seeing organisms that were before not accessible for staining work, simply because they have complex shells or something like this. And we're also seeing cellular organization that we couldn't record before simply because of amongst others, of the sizing. And so here the colleagues from the expansion microscopy team were very excited because they saw that there's the Basque microalgae culture collection. They've been working with the colleagues from there to actually receive cultures that they can preserve then on site and then work them off and generate this atlas of cellular structures. And so far they have, in the project of the expansion microscopy, they've fixed around 300 species from multiple culture collections. They're trying to cover as widely as possible the unicellular eukaryotes diversity. And then they're working off all the stainings from more generic all the way to very specific things, and to really get a good a cellular atlas of unicellular plankton. Now, Yvonne mentioned the 3rd pillar of TREC previously, and I want to also reiterate this here. It's not only about science, it's not only about working with you, but it's also about outreach. So a big part of the whole expedition is also public outreach and public engagement. Throughout all of these countries, my colleagues from the outreach team are actually engaging in the local language with everything from school kids to interested public to not yet interested public through a diversity of different ways. They've generated a game for kids to play and to learn about biodiversity and biodiversity loss. There is mobile exhibits, there's a science on tour which will happen tonight, and as well as visits to our mobile lab, there's visits to Tara, and also working with local schools to spread this message. And so again, only echo what Yvonne said earlier. If you're interested, if any of this, what I just told you, excites you, please have an eye out for these activities over the next week. So we started in March. Where are we so far with our whole expedition? We've done quite a few kilometers, starting in the marine station of Roscoff, going all the way up to Estonia, and then via the Nordic countries and the UK back to France. The advanced mobile lab is already here, and the colleagues from Tara and the smaller lab will arrive actually today, and then for the next week it's science, science, science. We stopped at about 20 of our partner institutes so far. We are currently performing around 200 different protocols on each site. We collected about 20,000 samples already that are all then first shipped to a central sampling hub in Germany, and then from there also distributed again to collaborators throughout the project. And as you see, it takes a village to build up something like this. This is just a little overview of all the people that are involved, from thinking about this project, executing this project, all the way to the people in the field that are actually doing the science, all the people behind the scenes that are taking care of the legal aspects, the logistics aspects, and so forth. And of course, all our collaborators throughout this expedition, from the sampling coordinators that informed which ways would be best to sample, to the Tara team, our advisory team, and so many, many, many more people. And with this, I would also like to thank you for your attention, and to the both of you for giving me the opportunity today to speak about our project. Thank you very much.
Okay, I think it's quite exciting, and if it's so exciting, there should be some questions from the audience. Do you have questions? Come on, it's about science, and it's about traveling. There should be questions in the audience.
So far, do you have any conclusion about all the data you have collected on the north of Europe?
Sorry, could you?
So far, do you have any conclusion about all the information you have got in the north of Europe, or have you done an analysis?
So. Yes. So for the big overall data sets, we're still in the collecting phase. It runs this year until November, and then next year we're covering the Mediterranean. And so if you really want to have a look at the big picture, then of course we need to finish first the collection before we can dig in. However, smaller of the plugin projects already are generating data. Some of this I've showed you today with the beautiful images from the plankton expansion microscopy. Collaborator emailed Ibon and me yesterday from the first data set from Roscoff, and we both had a little ear in our eye.
We received a video at night, at 10 o'clock at night yesterday, and myself, I was crying.
So the smaller projects are already churning out results. Of course, they're also always interested in having a perspective over the whole of Europe. But of course, you cannot not look once you get the first results.
Don't be shy. I understand Basque, I understand Spanish, so I can translate.
You have briefly mentioned that there's activities that are happening at the same time that you're investigating, that you're researching. What exactly are those activities? Because we have heard that there's workshops and things like that in Plencia next week, and just to know a little bit what that is about.
Maybe I show them?
Yeah.
You can go to the web. You can go to the web page of the Plencia Marine Station, and you will find that there is a section on the TREC expedition. And there is a calendar of activities. It's in the 3 languages. So for instance, we have an event tonight. come here to the web page of the Plencha Marine Station, you see here it says TREC expedition. If you click there, you have all the explanation of what is TREC, and down here you have a calendar of different events. So last Friday it was the European Scientist Night, and we were already there. 1st of October, Sunday, we had a estuary cleaning event that's already passed.
Today there might be a talk.
Today, it's his talk, but we are also having science on tour. We are having, it's a kind of a pint of science, yeah, but it's going to be in the Ateneo in Bilbao. So if you want to see Nico again, he will be there, but talking more informally. And very importantly, 5th of October, we have open days. We have open doors. If you want to see the advanced mobile app, it's going to be open doors tomorrow. And it's going to be open doors for the marine station tomorrow. So in the morning is for schools, in the afternoon is for citizens. And you are citizens. So you could register there if you want to come tomorrow to the beach. Yeah? And there are more things. Just check. The Tara boat, which has just arrived in Bermeo. My son is studying in Bermeo, and he has sent me photos. It's in Bermeo, will be docked in the Chas Museum in Bilbao for 7 days, and it will be open for visits. So that's the calendar of outreach events, and you can participate if you want. Yeah? Yeah.
Okay, as we are talking now about the outreach, we need help for the outreach. We need volunteers. We are going to be the outreach during the bank holiday that we have. Okay, now? Yeah. The person in charge of the outreach, the outreach is not here, is Eider Bilbao, but she has asked me to ask for volunteers. As you see, the events are during the bank holidays that we have, through the 12th, 13th,
14th, 15th, and 17th of September. Yeah, most of you will be on holidays.
Yeah, but probably
what else
some.
Can
Yeah.
you do better on holidays than doing a little bit of science?
So probably.
Yeah.
You find so probably you will be here as well. If you want to take part disseminating science, knowing the researchers that are involved in this TREC expedition, and what is more important, disseminating science, you can help us. We need translators for all the activities, because the citizens, they do not know French, German, or English. So we need translators to Basque or Spanish or to any other language. If you are interested, you just write an email to eider.bilbao@ehu.eus. Eider Bilbao probably she has been your teacher. Write the name, your interest, the language that you normally use, what are you able to translate, and the day that you are free for doing this. And she will contact you, right? That's it. Oh, I do have a question for Nick. I've been teacher of many of the students that are here, and we normally talk about the expensive that science is, how much it costs. Can you give us an idea of the cost of the TREC?
That's a forbidden question.
Just an estimation.
So with everything installed and what not, it's about 2.5 million euros. Sounds a lot when you hear it like this. If you look at what marine research and marine research vessel costs, so I think we're a little bit cheaper than them.
Do you understand what's the EMBL, the European Molecular Biology Lab? Explain them.
Okay. So we're actually celebrating our 50 years anniversary at EMBL, and EMBL was funded 50 years ago by the idea that Europe should unite to ensure that research happens also in Europe as a bit of a counterpart to the American Ivy League universities. And so there was the drive by some very clever and some very good people to unite the European countries to pledge funding to have a European-owned research institute. And so it's actually, I mean, yes, the headquarters are in Germany, but it's literally the most diverse place that I've seen. We have colleagues from all over the world. There's so many languages spoken on a daily basis on the institute, and so the idea is really to have a truly European research institute dedicated to excellent research on the first hand, as well as building communities, teaching, training, and serving the European research community. We have a ton of courses if you're interested in pretty much anything in terms of biology. Maybe not field sciences yet, but they are about to come. But anything in terms of molecular biology, there is courses around the whole year round. We have a whole building just dedicated to all our courses. We have the core facilities that are accessible for collaborators if you're interested in running a research project there. And now with TREC, we're also in your country, happy to do science with you.
And they have, in 50 years of history, they have 3 Nobel Prize winners, which is quite a thing, eh? No more questions? If you come tonight to Ateneo, we can talk through a beer. Yeah?